Welcome to Azad Bioacademy. Today's lecture topic is pharmacogenetics and pharmacogenomics. Okay, so firstly, uh, before going to details, we have to know the basic things about the pharmacogenetics and pharmacogenomics. So firstly, come to the terminology pharmacogenetics. So if I just explain it into two terminology, one is pharmacon, another one is genetics. Okay, pharmacon means drug, and genetics, gene related matter. Okay, so if you think about the pharmacology and the gene, pharmacology is study of drug and body interaction, and genetics, the genetic materials, gene, basically you can say the DNA. So drug and gene relation, what type of relation it is? That means when your gene is responsible for altering the function of drug, altering the activity of drug, that is the pharmacogenetics. So gene means when the single gene will be responsible, that can reflect the terminology pharmacogenetics. Okay, that means a gene is responsible for altering the function of drug, that is the pharmacogenetics. Okay, and this terminology, the pharmacon or pharmacology, is the same terminology, and this is the genomics. That means this term comes from genome. So what does it mean, the genome? Genome and gene. Gene is the singular unit, particular part of the DNA which is responsible for particular function, or you can say which can express, that is the gene. So a single part of the DNA which is responsible for a particular function with carrying some particular sequence of the DNA, that is the gene. You can say part of DNA. And genome, you can say in different way, that is more than 1 to 3 gene, you can say genome. That is multiple gene, you can say genome. You can say in different way, that is entire genetic material within the cell or in your body, that is the genome. Okay, so pharmacogenetics, when a single gene is responsible for altering the function of the drug, that is pharmacogenetics. And pharmacogenomics, when multiple gene or more than one gene will be responsible for altering the function of the drug, that is the pharmacogenomics. Okay, so gene and genome, singular and plural, that would be the best best thing to just discuss about it or understand about it. Okay, so without actually giving example, it is very tough to understand. So come to the point that pharmacogenetics will be explained by giving an example. So here it is, this is the example of pharmacogenetics. So this is the drug, that is clopidogrel. This drug is antiplatelet drug, and this drug is basically considered as a blood thinner, as like aspirin. Okay, so clopidogrel is actually a prodrug. It's a prodrug. So the prodrug is actually the which drug is taken in a inactive form, and after entering into the bloodstream, then you can turn into active form. That is prodrug. That means you have to take it in active form, and after reaching to the systemic circulation, then it will be active. So this is the prodrug. So that drug must be altered by metabolism, then it will turn into active form. So which enzyme is responsible for uh, uh, making the changes of this clopidogrel structure? So this is the gene which is responsible uh, for encoding an enzyme. That enzyme is responsible for metabolism of that clopidogrel. So this CYP2C19, this is a gene. Okay, this is gene, and that gene will express to produce a particular enzyme. You can say this name of the enzyme, that is CYP2C19 enzyme. Okay, so that enzyme is responsible for altering the inactive clopidogrel into the active. Okay, so in generally most of the clopidogrel will be in active form by that enzyme. This is the normal phenomenon. Okay, you will take the clopidogrel as inactive form, and that enzyme which is encoded by that particular gene, then it will turn into maximum amount is 2-oxoclopidogrel. That's okay, and certain amount or little amount can turn into 8-oxoclopidogrel, that is inactive form of the clopidogrel. Okay, so no problem. When any alteration occur in the gene of CYP2C19, that gene will be mutated or alter the DNA structural the gene structural changes. Okay, that is mutation, base alteration or deletion or addition. That means the sequence of DNA will change, particularly to produce a altered protein. Okay, so when that gene will be mutated, then the scenario will be changed. What will happen? 85% clopidogrel will turn into 8-oxoclopidogrel. That means inactive by the mutation of that gene, and only 15% will be active form, will be in active form. Active form. Okay, that means maximum drug will be inactive. So you don't know what is happening within your body, because without the study of your genetic expression on the drug, then you don't know what will happen in your body. You know, okay, I'm taking the clopidogrel and it will work in the normal way, but you don't know your particular gene is just encoded a particular enzyme, and that is already altered, and altered enzyme modified or some changed enzyme, that is just changing the pattern of clopidogrel maximum into 8-oxoclopidogrel, and you are expecting I am taking clopidogrel and it will work properly, but actually happening in opposite, and you will suffer a lot. That means you will not get the proper cardiovascular protection, that is blood thinning activity of the clopidogrel. So that is very much important in individual people. Clopidogrel will express and how way. That means this enzyme will work, or the gene will how it will behave on the drug. That will depend on the person to person. So you have to study the gene and its effect on the particular drug. So this is the pharmacogenetics. Okay, so man to man, as it is just changing the activity. So if you just prescribe a particular medicine for a particular person, so this is the personalized medicine, this is the precision medicine, and that way if you just get the information, then you will prevent the maximum side effect, or you will get the desired activity. That means you will get the maximum activity of the drug. Okay, so this is the example of pharmacogenetics. That means a single gene is responsible for altering the metabolic pathway that was very much necessary to make the clopidogrel from inactive form to active form. But after mutation, the scenario has been changed in completely the opposite way. That is 85% of drug has been inactive. So this is the matter of pharmacogenetics. Okay, so now come to the point to the pharmacogenomics. So already I discussed about that. That is pharmacogenomics. That means multiple gene will be responsible for altering the function of the drug. Okay. So come here. The two gene is one is CYP2D6 and another one is CYP2C19. Both genes are responsible for the metabolism of amitriptyline. So this is the amitriptyline. This is antidepressant drug. You know, this drug is belongs to this drug is just considered as a tricyclic antidepressant. Okay, so this antidepressant drug will be metabolized by two types of enzyme comes from the encoded uh, product of that two particular gene. That means two gene is responsible for the production of two enzyme, and that two enzyme will be involved in the metabolism process of the amitriptyline. And that those genes will just give its function on person to person in different way, because the genes which are responsible for altering the metabolism, that has various DNA sequence. That means the DNA sequence can vary. It is naturally located gene which can vary its sequence, as like you know the terminology, that is allele. Particular location, a particular DNA is gene is present which can change its sequence. That means can vary the sequence, and then slightly the activity will change. That is the matter. If you think the previous example, the mutation occurred on the gene which is responsible for the alteration of clopidogrel, and here mainly alleles are responsible. That means various DNA, various gene sequence, the variation of the gene sequence is responsible for altering the enzyme activity, and that enzyme activity when will change, that will change the metabolic pattern of the amitriptyline. So what is the problem? The problem is that in my body such type of expression is just happening, but another person no problem at all. Another person has another problem. Another problem means actually some metabolic rate. Suppose in my body the metabolic rate of that amitriptyline is slow or very poor because of the expression of that enzyme, and to another person the metabolic rate is very quick and very high. So the dose would be the same or should be the same for amitriptyline? No. So before prescribing the drug, if you do have the genetic information and expression pattern, so you can easily prescribe the exact amount of dose and exact the best fit drug for that person. So that uh, information when you will have, then you can easily prescribe for a precision medicine for a particular person. So for the discovery of the drug, for the clinical trial, or to remove or to minimize the side effect, adverse effect, or to get the more efficacy of the drug, or particular dose, then you will just get a particular desired activity of the drug. So that is the beauty of pharmacogenetics and pharmacogenomics study. And this age, modern age, is just going in that way, that although throughout the world it is in initial stage, but we are moving forward to establish the precision medicine or personalized medicine after discussing or after just studying the pharmacogenetics and pharmacogenomics study. So I will just sum up the today's discussion. That is, pharmacogenetics means a particular gene is responsible for altering the drug function or drug activity, and when multiple genes will be responsible for altering the drug activity, that is pharmacogenomics. And already I have given two examples. One is clopidogrel by a single enzyme, a single gene, and another one is amitriptyline by two gene. So this is the end of this today's discussion. If you do have any question, then please ask me in my comment box, and any suggestion you can give me, that will be more helpful for me to develop my content as well as my confidence. So thank you very much.